الجلسة الثالثة. فليتفضل كل من الدكتور سعد الصعب والدكتور بهاء الدين حسن والدكتور غلام مصطفى. يتفضل الطالب فهد السهلي لتقديم البحث الأول.
بسم الله الرحمن الرحيم. السلام عليكم. My name is Fahad Abdullah Alsahli. I'm third year of medical student at Doadmi College of Medicine at Shaqra University. My topic is based on burn research, and as we know, burns occurs when some or all the different layers of skin of cells in the skin are destroyed by a hot liquid or a hot solid, which is contact burns or a flame burns. So burns pose a serious global public health problem with over 100,000 of deaths annually from fire-related burns alone. As I said before, my topic is about isolation and identification of burn skin pathogen and their antimicrobial pattern. So the morbidity and mortality in hospitalized burn patients is due to infections, and due to these infections, the 75% of deaths arise by thermal injury, with burn in 40% of body surface area. The serious thermal injury causes total loss of the skin surface over a large area of the body, as it acts as a barrier to a microbial host invasion. It's not surprising that the risk of subsequent burn wound infection and systemic infection depends on the size of the burn injury. And due to the loss of the outer layers of the skin in a severe burn, there is marked loss of body fluid and danger of secondary infection with gram-negative bacteria such as Pseudomonas or with fungi such as Candida albicans. Secondary infection means invasion by a new organism of tissue damage by an earlier infection. The patient with burn injury are at high risk due to the prolonged stay in hospitals and intensive diagnostic and therapeutic procedures. A lot of research has been done on isolation, characterization, and identification of the burn pathogens, but still more to be done in this area. So our research aimed to collect the pathogens from burns patient and then isolate them to identify the pathogens. Then finally, assessment of antimicrobial pattern of these pathogens. So 50 bus samples were collected from burn patient with invasive wound infection using sterile cotton swabs. Specimens were inculated aseptically in nutrient agar, mannitol agar, salt agar, Pseudomonas agar, and McConkey agar, citramide agar, and Acinetobacter medium, Euseum methylene blue agar, and blood agar medium. Following inoculation, all the culture plates were incubated at 37 Celsius for 24 hours to 48 hours. In an inverted position, pure cultures were recovered by repeated subculturing. Antimicrobial percentage of resistance pattern of Pseudomonas, Staphylococcus aureus, and E. coli, and Klebsiella against ciprofloxacin, amikacin, cifeprime, gentamicin, nitrofurantoin, imipenem, meropenem, fluoxetine using agar disk diffusion method. So here's the result was done by the using of acid production test, and this test was done to be found for these bacteria. As we know, as we see here, like all of them are gram-negative except Staphylococcus aureus. So here we can see the most common present the pathogens in the burn tissue is Pseudomonas aeruginosa. Then followed by which represent as 39.5%, followed by Staphylococcus aureus with 25.5%, then Acinetobacter pneumoniae for 10%, and Klebsiella for 0.2%, then Protease vulgaris 3.5%, and finally E. coli for 2%. The sensitivity of Staphylococcus aureus was scored towards imipenem and meropenem, and partially sensitive to fluoxetine. E. coli was found to be 100% sensitive to ciprofloxacin, and Klebsiella was found to be sensitive to amikacin and gentamicin respectively. So we can conclude that the pathogens were isolated and characterized from 50 burn patients. The most preferred location was where the upper and lower extremities, and their antimicrobial activity pattern was evaluated using disk diffusion method and ciprofloxacin as a standard drug. The isolated pathogens were Pseudomonas aeruginosa, Staphylococcus aureus, Acinetobacter pneumoniae, Klebsiella, and Protease vulgaris E. coli. Most of the pathogens were found to be drug resistant, while several isolates were noted to be multi-drug resistant. The growth of multi-drug resistant organisms should be considered as a serious risk factor in a burn unit. Aggressive infection control measures should be applied to limit the emergence and spread of multi-drug resistant pathogens. So here's my reference. Thank you. So if you have any questions, I will be appreciated.
We can receive a question from the audience for the presenter.
I have one comment here. If we bring the discussion, you will find you wrote results in the discussion. Give me the discussion slide.
Yeah, I focused on it.
It is result.
Yes.
We don't need to write results in the discussion, only comments. No results in the discussion. That's one point.
Thank you. Any other question?
Any question?
Yes, please.
Prof?
Yes, Prof. Atif.
Thank you for this excellent presentation. I have comment in fact regarding the results. We have a sample of about 50 patients, and if you found a lot of bacteria infection in these wounds, and from these samples you get results that this type of bacteria is sensitive to certain type of antibiotics. So I don't think that this is considered sensitivity for certain type, because we don't have a big sample for one microbe. If you, for example, we have 50 samples for one microbe, okay, it is with high sensitivity to this antibiotic. But for 3 or 4 Klebsiella or whatever the organism, we can consider this antibiotic is sensitive and consider it as highly sensitive to this organism. Thank you very much.
Thank you so much.
Is the questions from behind? Yes. Welcome.
Congratulations for very nice presentation, student. Why you have done this study?
Why?
Why you have done this study?
Why?
You have done this study.
What was the objective?
Because there is 75% of death arising from thermal injury from burn patients. So that's why we chose to this research to be done. Thank you.
I'm sorry, just missed the first part of the presentation, but I want to know how the multiple drug-resistant strains were defined. What was the criteria to say this bacteria is multi-drug resistant and this one is not multi-drug resistant?
Okay.
What was the criteria?
The criteria were with another.
I'll just give you an idea to say if I choose 7 classes of drugs, if I say if from each class a pathogen is resistant to at least one member from 3 classes out of 7 which I chose, I will consider this as multi-drug resistant. So what was the criteria here?
The drug was used in this research was ciprofloxacin as a standard. So that's why we use this for.
And like choosing a single class, suppose you say ciprofloxacin, which is belonging to the fluoroquinolones group. Checking the sensitivity and resistant to only one class or one member of a class, you cannot say this is multi-drug resistant or extensively drug resistant.
Yes.
For that, one has to choose 7 or 8 classes of drugs, fluoroquinolones, aminoglycosides, cephalosporins. Then each class has different members, ciprofloxacin, levofloxacin, and from each class, if there is a specific number of drugs are resistant, only then we can say. So I think, was there any criteria used here or not?
Actually, it was there. Another drug from ciprofloxacin.
Is there a co-author available? If the co-author is available, or any senior person who was associated with him is available, he can also answer.
Dr. Barsana.
Yeah, to his question, and we have started to analyze the most commonest bug associated with the burns injury, and it was proved to be Pseudomonas. And we collected the data from the Dawadmi General Hospital, and which basically does the analysis as per the CLSI guideline, and they do as per the Kirby-Boyer method, and also they analyze the isolates with MIC. So whenever they analyze this, based on the MIC value, minimal inhibitory concentration value, they brand them as the resistant bug or the sensitive bug based on the clinic CLSI guideline. So we found ciprofloxacin to be little bit effective against Pseudomonas. It's an observatory study, but though this group of drug may prove to be ineffective against Pseudomonas, and this was our observation based on our study, but though the sample size is very less, we cannot statistically prove that ciprofloxacin can be given to all the Pseudomonas burn injury.
His question is that how did you define the multi-drug resistant?
Yeah.
So did you get the answer? So what was the criteria? There is one thing that you labeled them resistant. The other question is, how did you define the multi-drug resistant?
Multi-drug resistant to different classes of drugs like aminoglycoside, ofloxacin, penicillins, cephalosporins, all these group of drugs. Multi-drug, as you know, I think I'm to the point, different classes of drugs.
I think we are getting deviated from the topic. As Dr. Shabir said that he was out in the preliminary session, first session, related to his presentation. Multi-drug resistance was not being the primary concern in his title. If we go to his title is primarily associated on the isolation of that, which type of microorganisms are there. So one, you should focus on that, you should answer like that. The second thing is, I'm asking you are the principal investigator because he is trying to defend you.
Yes.
Did you use ATCC guideline, ATCC strands using as a standard? Because then you cannot say until and unless you do not compare it with the standard microorganism, and it's because possibility of the resistance in the genetic makeup changes is also there. So I want to see whether you use that or otherwise, because that cannot reach to the conclusion.
Being his co-guide, so I take this responsibility because I have a close association with the microbiologist in Dawatmi General Hospital, and they are using the ATCC strains to confirm it is a multi-drug resistant bug. Thank you, sir.
Thank you.
Thank you. شكرا فهد.